Hello, Assalamualaikum. My name is Muhammad Fazlin bin Muhammad Fadli. I will present an introduction to the topic of mutation breeding in plants. It is a self-fertilization or selfing of mutant until it induces desirable traits that are needed. Mutation breeding is based on the induction and identification of mutation. The result for this method is fast, cost-effective and durable, non-hazardous and eco-friendly. Plantation culture method useful in order to reduce time to create a breeding mutant line. Mutagenesis is important to improve crop and free from regulatory restriction on genetically modified organism or known as GMO. Thank you.
History of mutation breeding in plants. The first known variations in plants was detected in China around 300th century. Back then, they were producing varieties of crops using hybridization technique. As time goes on, there were increasingly many types of plant varieties that had been produced unknowingly. Soon in 1868, a botanist biologist named Charles Darwin discovered mutations in plants. Darwin's botanical work was important to the development of his ideas on evolution and natural selection. Darwin was fascinated by the observation that a bee orchid, commonly to England, Ophrys epifera, was apparently adapted to self-fertilization. His further observations convinced him that these self-pollinating orchids still retained the mechanism needed for pollination by bees. Where he imitated a bee's action using an object, the pollinia reacted as in other Ophrys, readily attaching to what would have been the bee's head. He concluded that Ophrys epifera must have at one time been commonly pollinated by bees, but due to an insufficiency of pollinators, it became slightly modified so as to fertilize themselves. Arke Gustafsson, father of mutation breeding. Soon in the late 1920s, Ake Gustafsson and Herman Nilsson Ehle started experiments of induced mutation at the Svalöv plant breeding station in Sweden. Already in the mid-1930s, the first viable mutation appeared, and in 1940 an extended research program was set up. Gustafsson devoted much of his scientific career to mutation research. With funding granted by the Swedish government, he established a large national research group with the aim to investigate theoretical and applied aspects of induced mutation. In opposition to many contemporary geneticists and plant breeders, Gustafsson never doubted the value of induced mutation for plant breeding, which according to him dealt with the artificial evolution of crop species by changing and accommodating them to human needs and demands. He was dubbed the father of mutation breeding. Mutation breeding can be described as purposely application of mutations in plants. Unlike hybridization, mutation breeding is induced by the desirable character and exploiting it the new crop improvement. Mutation breeding draws attention in farmers because the technique can be used for developing the elite breeding lines and varieties.
Mutation is heritable changes in genetic material of living organisms, and it may be in spontaneous, which is natural, and some of them may be in induced, which is artificial. For the spontaneous mutation occur without intentional human intervention, and their causes are still unknown. Nowadays, currently, about 3,220 are officially had released mutant cultivar in 210 plant species, which is the majority of mutation is used for the plant breeding and for the crop production, and some of them is used from the treatment of physical mutation.
Experiment in mutagenesis and mutants. The first one is induced mutagenesis. Usually, induce mutagenesis, we will use radiation or chemical mutation by select the targeted mutagenesis and mutant plant or the organism that is produced by indeed mutagenesis that is referred to induce the mutants. It is either induced by mode of plant propagation, which is perturbation in gammatic, or either in somatic biological mechanism such as natural physical conditions like sunlight, UV light and others, by chemical agents such as alkylating agents and biological agents. The second one is insertion mutagenesis. DNA insertion by genetic transformation and insertion of tDNA, which is tDNA is insertion mutagenesis or activation of transposon elements. The third one is site-directed mutagenesis. The process is by creating a mutation in DNA molecule.
Among the current issues that can be linked is plant biotechnology. Plant biotechnology is important in mutation breeding, which is plant tissue culture technique. Plant tissue culture requires a short time to generate a breeding mutant line, which is passed down from curtain traits into its offspring. Any crops such as seed or vegetatively propagate crops and impossible crop of crossbreeding and transgenic origin, as well as any initial material from the plants such as seeds, pollen seed, cutting or tissue culture, as well as individual wool plant, can be mutated using mutation technique. Another method from plant breeding is the double haploid technique, which is involve doubling the number of chromosomes in haploid organism with only one of each chromosome pair. There are several techniques that have been developed and can be used to expand mutagenation in population and the diversity by identification of mutation. One of the methods used in denaturing HPLC known as a chromatography technique that can supply different homodiplers and heterodiplers, if not mismatched. If it was used to detect single nucleotide polymorphy in the human white. However, the most instruments have only HPLC column. DHPC has major constraint in the terms of the number of samples that can be processed at the same time.
So the other current issue is in vivo and in vitro propagation. So somacrona variant may arise when repeated round of propagation, and it's depending on the nature propagation that can be classified as protoclonal, gammaclonal, and caliclonal. So in vitro culture is combined with induced mutation that can speed up the reproductive process from the variability creation through selection and also replication from the target genotype. So it is appear in recent improvement that based on this technology is most likely provide a better method for the selecting selected mutant. So other than that, while tubers, rhizome, stem cutting, and also seed has common in vivo propagation method that significant development in vitro culture that have result in enormous advance in the horticulture crop micropropagation. So some restriction in the application of mutation approach that involves seed and vegetatively produced plant that can be solved by using in vitro technique such as entoculture, shoot organogenesis, somatic embryogenesis, and also protoplasm fusion. So the other current issue is irradiation provide heritable genetic changes. It is large-scale deletion, inversion or transcolation of chromosome or other point mutation, which is form of mutation that produce a single modification, insertion or deletion of the genetic material in the DNA. It is whether is spontaneous or induced that has common source of mutation. So for the physical mutation that usually produce chromosome alteration and bigger in the DNA deletion, whereas mutagenic chemical that usually result in a point mutation. For the degree in mutation that can be varied, such as time of exposure and dosage of the tissue that have been exposed. Next, the ionic radiation is one of the physical mutations that can increase the rate of the natural mutation and have been widely used in induced heritable genetic changes. And for example, is gamma ray that have been commonly used as a mutagenic agent in the plant mutation breeding, seedling, or the whole of the plant that have been exposed to the gamma ray, and it will result mutation are not repaired by the cell itself.
Okay, I will continue the presentation. Uh, this is the future prospect of mutation breeding in plant. The first, it can improve high-yielding vegetable and seed crop that are being developed. For example, plant breeding necessitates the use of correct breeding procedure and technique, and it can improve the management practice and can maintain or increase plant quality and ensure pest and disease resistance. After that, pathogen with high levels of gene flow have more genetic diversity than those with low level of gene flow. The gene flow is particularly relevant in plant disease because it is linked to the spread of pathogenic mutant LS among various population. The genetic manipulation of population is raising the frequency of desirable LS in cross-pollinated crop, and it also introducing citrin males in self-pollinated crops such as wheat and rice. It is the one of the future promise of mutant breeding in plant. And lastly, the primary goals of transgenic plant was development is to generate crop with the optimal feature, high quality, and high yield. And the plant have been shown to be capable of acting as a factory for medicinal protein manufacture, and it also being advantageous to agriculture. And that's all from me. I will pass the next presenter is Hasbullah.
To sum it all, uses of mutation breedings are important to improve crop quality in terms of alteration of colors, form of seed, and taste of the fruit. With the presence of mutation breeding, variety of crop has been produced over 1,000 varieties. Hugo Marie de Vries are the one who responsible in genetic alteration, and he discovered the laws of heredity and introducing the words mutation. All in all, technology has been proven to bring benefits in genetic field, which benefits all mankind, especially towards mutation in crops. That's all from us. Thank you.